grade 12. Today we will continue our chapter and discussion about the immune system and the components of the immune system. And as you remember in document 1, we discussed the HLA proteins which are found on all nucleated cells of the body, and they are identity markers unique for every individual. Today we will be talking about the blood groups, which are another self-marker. And as you remember and you know from previous discussions in grade 8, that the blood groups in the ABO system are 4: A, B, AB, and O. Now, these blood groups were first discussed by a scientist called Karl Landsteiner in 1901. Now, this scientist discovered that the serum of some individuals can agglutinate the blood of others and might not agglutinate the blood of other individuals, depending on their blood group, and that's when the talk about blood groups started. As you know, when we discussed genetically the blood groups in genetic from a genetic point of view, we said that there are 4 blood groups: A, B, AB, and O, and these blood groups are characterized by the presence of a substance H, which is basically a carbohydrate, and on which we have a certain marker attached due to the presence of enzyme A, enzyme B, or the absence of this enzyme. And we said that galactose plus substance H gives us blood type A, and acetyl-galactosamine plus substance H gives us blood type B. The absence of any of these sugars gives us blood type O, and the presence of both gives us blood type AB. Now, from an immunological point of view, we can discuss the presence of the 4 blood groups due to the antigens or agglutinogens found on the surface of the blood cells and the antibodies or the agglutinins found in the serum. And blood type A has antigen A on the surface of the RBC and anti-B in the serum. Blood type B has antigen B on the surface of the red blood cells and anti-A in the serum. Blood type AB has both antigens A and B but no antibodies, while blood type O has no antigens but both anti-A and anti-B antibodies. By a simple diagram, we can see that, for example, if blood type A has antigen A, which we give here like the shape of a pyramid or a cone, the serum should contain anti-B, which cannot bind to the antigen on the surface of the red blood cell, or else agglutination would take place. And similarly, antigen B has a spherical shape, and thus anti-A cannot bind to the antigen found on the surface of the red blood cell. So type B has antigen B and anti-A antibodies, but type A has antigen A and anti-B antibodies. Now, to elaborate more on this, we can explain how to test for blood type. So we have a slide on which we add 3 drops of blood from the individual to be tested, and we add anti-A, anti-B, and anti-H. Anti-H stands for rhesus factor. Rhesus factor means positive or negative, and we will talk about this later on. For example, if I add anti-A and then agglutination takes place, this is agglutination where the cells come close to each other, forming like spots. But with anti-B, nothing happens. This means that this person has antigen A on the surface of his red blood cells, and so he has blood type A. The anti-H also forms agglutination. It means that it is A positive. It is rhesus positive. And we will soon talk about what is the rhesus factor. For example, this person here has agglutination with his both in the presence of anti-A and anti-B, and this gives us a blood type AB, and since anti-H makes no agglutination, then he is AB negative. Also, for this individual, for example, no agglutination takes place in all the 3 drops of blood, and thus he is O negative. If only here we have agglutination, then it is O positive. If only here we have agglutination, then it is B negative. So depending on the agglutination with the antibody, we can know what is the blood group. Now also, why does agglutination occur when we transfer blood, and who can give who and who can receive blood from which blood type? Also, this is very important to understand. For example, here we can see that the donor, which gives us the red blood cells, and the red blood cells of the donor are being attacked or detected by the plasma or the serum of the recipient. This is the recipient, and now if for example, we have the donor giving us blood type A, and the recipient has anti-A in his serum, then the anti-A would just connect the cells and agglutinate the cells in this way, and thus we will have a process of agglutination, and thus the blood transfusion will be rejected and it is not accepted. And thus we can start to think which blood type can give which individual and how. Okay? Now also here on your book you can see the blood test. This is the presence of agglutination. This is the absence of agglutination depending on the blood type. Also here we can see the agglutination and the rejection, where the antibodies can agglutinate the red blood cells, but if no agglutination takes place, then there is no rejection of this blood. Now what about the blood transfusion? The transfusion chart, as we see here, blood type O can give A, B, and AB, and for sure can give blood type O, because as we know, O has no antigens on the surface of the red blood cells, and so it can be accepted by all other blood groups. However, A can take from O and give itself, because O, as we said, has no antigens, and A has anti-B, so it cannot receive from B or from AB because they have both antigens B. Similarly for B, which has anti-A, and AB, which has no antibodies, can receive blood from all the types, and since it has both antigens, it can only give blood to itself. Now finally, what about the rhesus factor? What do you mean by positive and negative? So the rhesus factor, and this here we have an experiment. First of all, the scientists collected blood from a monkey called rhesus monkey or macaque rhesus monkey. They took out the RBCs, extracted RBCs from this animal, and if we say that we have 100 individuals giving blood or RBCs, then we remove the serum from a rabbit. This serum of the rabbit cannot agglutinate the blood of the monkey nor of the two types of individuals or of the 100 individuals, because the serum of the rabbit does not have antibodies against the RBCs of the monkey or of the human being. However, if in a second experiment we take or extract the blood from the rhesus monkey, injected them in the rabbit, and then we took out serum from the rabbit, this serum will agglutinate the blood of this monkey because it has antibodies now. It developed antibodies against the blood of the rhesus monkey. But our surprise was that 85% of the individuals, of the humans, their blood was also agglutinated by this rabbit, although their blood was not injected into the rabbit, which means that the rabbit has developed antibodies against the monkey. The blood of the monkey could also agglutinate the blood of 85 individuals, but could not agglutinate the blood of the 15% of the other individuals. This means that the RBC of these humans shares at least one protein or one antigen with the RBCs of the rhesus monkey, and thus the antibodies which bound to these antigens here could also bind to these antigens on the surface of the red blood cells of the humans and agglutinate them, and that's why we refer to these humans as rhesus positive, because they share at least one antigen with the rhesus monkey, and that's how agglutination took place. So this is the second document of our chapter, which is about the blood groups, and I think it's very easy, it's not that difficult. The third document will be about the self and non-self and will be discussed later on. Thank you.